welcome back everyone. I think we'll make a start. So I'll just run through the learning outcomes and then we'll do the attendance pin. So these are the learning outcomes for this lecture and also tomorrow's lecture. So obviously this week we're going to look at the Immunologist toolbox. So by that I mean all the techniques that we can use sort of in an immunological setting, but also a lot of these techniques, as you will find, have actually sort of gone beyond the realms of immunology and are quite widely used in many labs and research settings. So at the end of this week, you'll be able to understand the types of experimental models that are commonly used to answer complex immunology questions. You'll also understand how to prepare and analyze leukocytes, so white blood cells, to be able to use antibodies as research tools, and also get an understanding of flow cytometry or FACS analysis, which is obviously a powerful tool for understanding and dissecting the immune system. Be able to define gating in flow cytometry, as well as understand size and granularity of cells, and also how to read different types of flow cytometry plots. So this is tetramer staining plots, surface markers, activation states, as well as cytokine secretion dot plots. Now, as I mentioned, the Immunologist toolbox, this is actually valuable to understand the types of experimental models that are available to us in immunology, but also in many other fields of life sciences research. And not only is this good as third-year students, but it's also good for your research projects, because I'm sure some of you will be encountering aspects of what we'll talk about today in one form or another. And of course, it's also good for any future scientific-based career that you may choose to embark upon. So I think whilst most of you are here, let's do the attendance pin. And the pin is 1592. So that's 1592 for any of you that didn't get it the first time. Okay. So first of all, I wanted to start off with the scientific method. So what is the scientific method? Well, essentially, it's this kind of circle of questions here. And this is pretty much the same scientific method that you will encounter for all sort of aspects of research lab. But essentially, you have a question or a research question that you're interested in. You then want to research your topic area, design a hypothesis, and then you want to test that hypothesis with an experiment. You then analyze the data and the results, and then ultimately you draw a conclusion. And then in most situations you would communicate the results, so whether that's by a presentation, a poster presentation, a paper, or a conference. And then this feeds in obviously to your final observation, but also perhaps to the next research question. And these are all important skills that are sought by future employers. Now, as I've said here, this case tends to be iterative. So what I mean is it's a cycle. You know, it's rather than being a straight line, it's this constant revolving cycle that feeds into the next question. So one cycle will feed back into the next round of question asking and the next research hypothesis that you'll come up with. So it's a constant feedback as you progress through your research project. Now, the scientific method is the experimental testing of a hypothesis, and it's formulated after the systematic objective collection of data. So that's important, that you want to be objective about it. You want to avoid any bias, so you may well have some personal biases if you're, for instance, researching a disease upon which a family member may be affected by that. So it's important to be able to leave out bias from the equation. And we've got a quote here where someone has said that I now appreciate how much I learned by being wrong. I can change my mind when confronted with a rational argument without the need to have the change appear to be purely semantic or hope that it will pass unnoticed. I'm grateful to be in a profession where the realization of being wrong is equivalent to an increase in knowledge. So it's really important to sort of have that unbiased approach, and be aware of that. Now, experimental techniques are essentially your toolbox when it comes to immunological research, but also your wider research as well. And learning the ins and outs of the methods will allow you to better understand the subtle sort of nuances in the field of immunology. So if we go back to this diagram, which is obviously the anatomical distribution of immune cells. So on the left here, you can see the immune organs as well as all the lymph nodes. So the first step in studying leukocytes, so leukocytes, white blood cells, is to isolate them from their in vivo location. Now, human lymphocytes can be most readily and easily isolated from peripheral blood, but then you have to address the question, these are accessible, but is it representative? So obviously blood is accessible, but many immune cells are also present at the body barriers, including mucosal sites, as well as other locations of the lymphatic system. So you have to bear that in mind, whether it's accessible or representative. So once they're isolated from the body, their behavior can then be analyzed in vitro, and then that also brings in one other question, sort of in vitro versus in vivo. How does that infect immune cells? Because obviously when you're looking at it in the body, the cells are surrounded by their natural environment. If you take them out and study them in vitro, then they're in a different situation, and what effect does that potentially have on their function? So there are just some things that you need to be aware of. And then obviously we come to in vivo models of human disease, and the most well-known one of this, mouse models. Now this is just an example of some mouse model strains that are available. So a lot of these are commercially available that you can buy and then bring into your animal breeding facility, and then you can base your model on that. You might well be familiar with the SCID mouse. So this is severe combined immunodeficiency, and that's actually something that we will talk about later on during the course of the module. So why use a mouse model? Well, we have this phylogenetic relatedness, and that we've got a physiological similarity to humans. One other main reason is we have this ease of maintaining and breeding them in the laboratory, because they have very quick breeding times. So therefore, when you're crossing strains and you're interbreeding them, you can do it very quickly than obviously with other animal models. You also have this availability of many inbred strains. So a lot of these are the ones that are commercially available. And when you have an inbred strain, this results in an animal that is genetically identical within each inbred strain. And this is really important because it means that your strain will be fundamentally free of genetic variants that could otherwise increase variation in your experimental results. So you need to bear that in mind. We also have the availability of strains with spontaneous mutations, so we can have strains that can develop human diseases. And then we also have an availability of humanized mouse models. So there's obviously quite a growing demand for specialized mouse models that have a human immune system. So what you can do is you can take your immunodeficient mice, such as a SCID mouse, and then you can engraft human cells or tissues so that you can actually construct this humanized mouse model. Then on the other hand, we have the questions of why not? So you might think size. Obviously, a mouse is a completely different size to humans. Also a different metabolic rate, but also more importantly, and I guess we're realizing that now, is they have a different diet, so therefore a different microbiome, also different pathogens. So you have all these different things. So you might have some convenient reasons for using a mouse model, but there are things that you need to bear in mind. And then of course we have ethics, but I think that's really beyond the scope of today's lecture. If we look at isolating primary immune cells from mice, we obviously have a range of different lymphoid organs. So we have lymph nodes, spleen, thymus, bone marrow, and peripheral organs. And so you can see here we've got a spleen and some lymph nodes that have been taken from a sick mouse versus a healthy mouse. So you can see the difference in size. So the lymph node accounts for about 50% of CD4 positive T cells, 10% CD8 positive T cells. So these tend to be mostly naive or memory. Spleen, we have 50% B cells, 25% CD4 positive T cells, and about 7% CD8 positive T cells. In the thymus, we have stem cells and developing naive cells. In the bone marrow, obviously, we have mostly B cells, so plasma cells, long-term memory cells, as well as lymphoid and myeloid lineages. And then we have the peripheral organs, such as the liver, skin, and gut. So you can see that obviously we can isolate different cells from different lymphoid organs. And then this is just showing basically an anatomical map of the primary lymphoid organs, but also the different lymph node sites that we have. Now, isolated cells can be analysed using FACS analysis, so some of you may know what this is, some may well be doing it in your third year project, but FACS analysis provides an easy way to be able to identify different immune cells, because obviously if you think about it, a T cell and a B cell, if you look at them under the microscope, they structurally look very similar. So you really need to look at their cell surface markers, so the CD4, CD8, in order to be able to actually accurately identify the different lymphocyte subpopulations. In terms of isolating primary immune cells from blood, so first of all, it's important to note that really there's a difference between plasma and serum. So plasma is essentially, if we take blood here and we heparinize it, so we treat it with heparin, and then you centrifuge it, what happens is you'll end up with a precipitation, you'll have plasma on the top, and you have your white blood cell layer here. So serum and plasma, they both come from the liquid portion of the blood, once all the cells have been removed, but that's really where the similarity ends. So serum is the liquid that remains after the blood has clotted. So if we don't add any anticoagulant and we spin it down, the blood will clot, whereas plasma is a liquid that remains once clotting has been prevented by adding an anticoagulant, and then we've separated out. So there's a difference there. But then if we look to how we might actually isolate peripheral blood mononucleocytes, or PBMCs, so this is how we would do it. So the NHS have a blood and transplant service, and you can actually get blood products for research labs from the NHS Blood and Transplant Service. So you can get what's known as a Buffy coat or an apheresis cone, and essentially these have been enriched for leukocytes during the blood processing once it's been donated. And you can request this from the NHS, and then you can perform density gradient cell separation. So what we have here is we've got a 50 ml tube, and we're layering our blood from the Buffy coat or the apheresis cone on top of a layer of histopaque or lymphoprep. So the names are interchangeable, brand names, whatever. But essentially it's a dense liquid. So you carefully layer it on top, and then you put it in the centrifuge and spin it for an extended period of time. And what will happen is it will separate out into something that looks like this. And so in your top layer you'll have your plasma. You'll then have a thin, white, fluffy layer of your PBMCs, so your peripheral blood mononuclear cells. You'll then have your histopaque layer. You'll then have a layer of granulocytes, so these are cells with more sort of like granular nuclei. And then you'll have your red blood cells pelleted right at the bottom. If you actually look at a photo of that, so this is a tube with just your histopaque or lymphoprep added at the bottom. The blood has then been carefully layered over the top, and then this is after spinning. So this is your PBMC layer right there, that white fluffy layer. So what you can do is you can then go in with a pipette and actually just carefully remove that PBMC layer, and then you can transfer it to a flask and then culture it or expand it. And we'll talk about a little bit more on that in the next slide. So PBMCs are any white blood cells with a round nucleus. So this includes T cells, B cells, natural killer cells, and monocytes. We then have our erythrocytes and platelets, which have no nuclei. And then we have the granulocytes, so our neutrophils, our basophils, and eosinophils. And these have multi-lobed nuclei. Okay, so now we've harvested our PBMCs. Well, what do we want to do next? Well, we can purify out specific cell populations, and these are sort of antibody-based techniques that we'll talk about during the course of today's lecture. So we can use immunomagnetic cell separation, and this can either be positive or negative selection. So positive selection refers to the process where we directly label desired cells for selection with an antibody or ligand that targets a specific cell surface protein, and then we can positively select them. Negative selection is essentially labeling all your unwanted cell types for removal with antibodies so that you're left with only the cells that you're interested in. So you've got two processes for doing that. Essentially, they both use antibodies against particular cell surface markers. We then have fluorescence-activated cell sorting. So this is based on sort of FACS analysis, but it's where you use a cell sorter, and this can actually be used to separate cells into specific populations. So you can harvest, let's say, just your CD4 positive cells or just your CD8 positive cells or whatever cell population you want based on the cell surface markers and the antibodies you use. You can also use immunodensity cell separation, which is largely based on centrifugation, like we've done over here. And this just kind of shows you a profile of the different densities at which you can separate out your monocytes, lymphocytes, basophils. But that's a little bit more crude, obviously, because you can see you've got overlapping regions between your different cell populations. But what we can also do is we can expand specific populations. So let's say we've got our PBMCs, but we want to isolate T cells or T lymphoblasts. So what we can do is we can take the PBMCs, put them in culture, and then culture them within presence of PHA. So this is phytohemagglutinin, sorry. And what this is a selective mitogen of T cells, and it will cause the T cells to become activated. And then if you add IL-2, well, we know that IL-2 is an important cytokine, so this sort of maintains and expands the T cell population. So you can get your PBMCs, and then with PHA and IL-2, you can rapidly expand the cell population so that you've got your T cell blasts. I then wanted to touch on transgenic mice. So ideally, this slide will move on to how we might generate a knockout mouse, and this is using the Cree-Lock system. But first of all, I wanted to talk about how we might create a transgenic mouse. If you want to create a transgenic mouse, ideally you would create a construct, which will be a plasmid or a vector that will encode your protein or perhaps a gene of interest that you want to insert into the mouse, so your transgene. And what you'll do is you'll transfect your construct into an embryonic stem cell. And then you will inject that embryonic stem cell into a blastocyst, and then that will be implanted into a pseudo-pregnant female. So a pseudo-pregnant female is one that's been given hormones to trick the body into thinking that it's pregnant, so it will accept the blastocyst. And then obviously you will get some offspring, and some of the offspring will contain cells that are derived from your injected cells that will contain your construct, and then you can breed your chimeric mouse or mice together so that essentially you create your transgenic mouse strain. Just sort of interbreed them to select for the traits that you want. So you obviously have to screen the mice for the presence of your transgene. So if we move on to knockout mice, so this is the Cree-Lock system. So let's say you want to study a particular target gene, so you can create that using the model on the left for creating a transgenic mouse. So let's say we've got a target gene here. What we want to do is we want to flank it with LOXP sites. So this is the gene that we want to knock out. So what we'll do is we'll create a transgenic mouse in which the target gene is flanked by these LOXP sites. And once we've got that mouse model up, that's brilliant, because then we can move to obtaining another mouse. And this is your Cree mouse. And some of these are made by other research labs, and some are probably commercially available. And you can buy them in let's say, a different set of different flavors almost. So what you have here is you have the Cree recombinase, but you have this under the control of a tissue-specific promoter. So let's say we want to create a knockout in T cells only. We will choose a Cree mouse that has Cree under the control of the LCK promoter. So LCK is a gene that's only activated in T cells. So therefore, LCK would normally only be activated in T cells. So by inference, Cree will only be activated in T cells. So what you do is you then breed these mice together. So you've got your LCK Cree mice and your mice that have the target gene flanked by Cree-LOXP. And ultimately, you'll create a conditional knockout. So a conditional knockout means that the mice will have a knockout of that particular gene in certain cells. So if we've had an LCK Cree mouse, we will have a knockout that's only in T cells, so in the thymus. So you can see here that in our target tissue, our target gene has been removed, so Cree has come in because Cree is only active in the T cells, and it has removed our target gene in our target tissue, so in this case the T cells. In all other cells, the target gene is unaffected because the CRE is under the control of the tissue-specific promoter. So that's how you would create a knockout, or a conditional mouse knockout. So these models are used extensively in vivo experimental animal models, and it's particularly important for studying the impact of a newly discovered gene. So if you knock out a particular gene in a T cell, you'll then ask, what is the phenotype? What is the phenotype of the mouse on a whole, but what is the phenotype on the immune system? So you might then want to study the different immunological subpopulations and find out whether knocking out a particular gene has any sort of immunological phenotype on your different cell subpopulations. It's also important in identifying regulatory regions of genes that are required for normal tissue expression, as well as determining the effects of overall expression in certain tissues. So this is what we talked about, the CRE recombinase for making a condition. And you can also make inducible knockouts. So these are ones where you can actually induce the knockout, so you can effectively turn that on or off. And that's useful for embryonic lethal knockouts that would otherwise be lethal, and you wouldn't get a viable mouse. If we turn now to adoptive transfer of immune cells, so in this scenario we have a wild-type mouse, which is CD4 double positive. We can then take the wild-type cells from this mouse, and then we can adoptively transfer them into a knockout mouse that's, let's say, is CD4 knockout, so it's immunodeficient, and then we can see, do we get a phenotypic correction? So do we correct the phenotype that is observed by this knockout mouse? Now, adoptive cell therapy has emerged as a novel treatment for immunodeficiency, cancer, vaccination, and it has a whole range of potential therapeutic applications. And the adoptive transfer of tumour-infiltrating lymphocytes, or genetically redirected peripheral blood mononuclear cells, is used to treat patients with advanced solid tumours, as well as patients with CD9 positive B-cell malignancies. So this includes various lymphomas and leukemias. But it's also important in autoimmune disease. So, for instance, the transfer of Tregs, or regulatory T cells, in order to treat type 1 diabetes, for example. Let's move on to antibodies and their use as research tools. So obviously antibodies are particularly useful. They're highly specific for their corresponding antigen. They're easy to isolate and study, and they're really invaluable probes of biological processes, as we'll see throughout today's lecture. We also have different isotypes, so different classes of antibody, and this can confer diverse functions, and they can be generated in large amounts as well. And of course, they have a very high affinity for their antigen. As I mentioned, a lot of the things we'll talk about today are widely used beyond immunology. So antibodies are quite readily used as research tools, and you'll probably be aware of some of these. So we can have confocal microscopy or immunofluorescence. So this is where you stain your particular protein within the cell, so you can use that on the confocal microscope. You also have immunohistochemistry, which is another microscope-based technique. You have immunoprecipitation or co-immunoprecipitation. So that's where you'll have a bead that's conjugated to your antibody that you'll use to pull out a particular protein from cell lysate. And you can also see, let's say you're pulling out the blue protein, you can see what does it co-immunoprecipitate with, so what does that protein interact with in your cell lysate. So that's IP and co-IP. Also Western blotting, which I don't have a diagram on here, but this is obviously where you take your cell lysate, you run it out on a gel, and then you use an antibody to probe for particular proteins in your cell lysate, and you can look for the difference in amounts of protein between different cell conditions or different cell types. You also have flow cytometry, FACS analysis, so that's here, and we'll talk about that later on. Magnetic activated cell sorting, which we touched on, but again, we'll go through that later on. And then ELISA and ELISpot, which we'll also talk about. So you can see that you've got a whole range of different techniques, and these are widely used beyond immunology. So the chances are that working in a lab, you will encounter at least one of these different applications, antibodies. So we continue to look at antibodies as research tools. So because antibodies demonstrate a high specificity for their target antigen, they're widely exploited as reagents for scientific research. And as we mentioned before, they're categorized as either polyclonal or monoclonal. And this really is dependent on the method of their production. On the left here, we've got polyclonal antibody. So you need to remember that these are heterogeneous. They're secreted by different B cells. So essentially, they recognize the same antigen, but they recognize different epitopes of that antigen. And they're generated by immunizing an animal. So this can be a mouse or a rabbit or a sheep or a donkey or anything like that. And then you purify the antibodies from their serum. And that will give you your polyclonal antibody. The advantages for using this, they're relatively easy to produce. You also get multiple epitope recognition, which allows you to get sort of amplification of your signal. But there are some disadvantages. So there's a finite supply, and there's lot-to-lot variability. So let's say you find a polyclonal antibody that works really well for your protein. Once all that batch from that particular rabbit has been used up, you have to then move on to another batch. And that antibody it will have generated a different immune response, and that can potentially affect the quality of the antibody. So that's another thing you need to think about. On the other hand, we have monoclonal antibodies. So these are homogeneous. They're secreted by one B cell clone. So they recognize the same antigen, but they only recognize the same epitope. So they're absolutely identical antibodies. And they're made by fusing an antibody-producing B cell with an immortalized myeloma cell in order to generate a hybridoma. Now, the advantages of using these is it produces a highly specific monoclonal antibody. It gives you consistent performance because there's no batches. It's all exactly the same, so you don't need to worry about running out of a particular batch. Unlimited supply, as I've just said. The disadvantages are that you have a longer development timeline because you need to generate your hybridomas, and it tends to be more expensive. So obviously the cost of your antibody when you buy it as a researcher would go up as well. If we look at how you would actually create a monoclonal antibody, so in this scenario, mice are immunized with antigen, in this case antigen A, and then they're given a booster, and then they're sacrificed, and their spleen cells, so the spleen B cells, are isolated. Now, spleen B cells can't grow in culture indefinitely. So what we do here is we take some myeloma cells, and we screen them first just to make sure that they lack this HGPRT, which is important in the future step. And then what we do is we mix the cells together and we fuse them using polyethylene glycol. And so what we do is by fusing them, we produce this hybridoma. And then we put the cells into this medium that contains HAT. And any unfused myeloma cells are sensitive to this because we screened the cells over at this stage. So any unfused cells will die, and then we're left with our hybridomas. And then we can select the hybridoma for its specific antibody. So we can specify that we want to clonally expand this one. So we can select it, and then we can get our cloned hybridoma, and this will produce our monoclonal antibody. And then we can keep that in culture, and then we can just harvest the antibody. So you can see that you can generate a large amount of antibody from your one particular clone. So we start to look at some of the antibody-based techniques. So first up is ELISA and ELISpot. So ELISA can come in several different varieties. You can have indirect, direct, competitive, or sandwich, and I'll show that on the next slide. So this is an example of a sandwich ELISA. So you have your antibody bound or absorbed to the surface, and then this will obviously bind to any target antigen. And then you can come in with a secondary antibody that is linked to an enzyme which will convert a substrate into a color change, so you can actually then measure that in a plate well format, such as a 96-well plate. So ELISA, it stands for enzyme-linked immunosorbent assay, and it's commonly used to measure antibodies, antigens, or proteins in biological samples. So it's important in, let's say, the diagnosis of HIV infection, pregnancy tests, as well as measuring cytokines in serum. So in this example here, you'd have your antibody, you would add, let's say, your serum, and if your serum contains your target antigen, it will be bound by the secondary antibody, and it will give you a colour change when you add your substrate. So it's important in detecting the presence of a protein or a ligand. Then we have ELISpot. So this is a similar process, but it's focused on measuring the quantity of cytokine secreting cells. So you have your tissue culture dish here. You have your antibody that's coating on the bottom. You layer on your cells, and if your cell secretes a particular cytokine that your antibody can detect, they will then be bound by the antibody underneath the site of the particular cells. And then you will wash them away and then do your various washes, incubate with a secondary antibody and so on. And what you'll end up is these spots depending where your cells were that were secreting your specific cytokine. So that's another application of using kind of like ELISA but on a cell-based system. And then this is just showing the four main types of ELISA. So you have direct ELISA, where you have your antigen which is bound by a primary antibody which is conjugated to your enzyme. Indirect, where you have your primary but you have a secondary conjugated enzyme. And then sandwich ELISA, and then competitive ELISA. So there's four different varieties that you may well choose to use depending on the particular research question that you have. And then we can move on to multiplexing. So ELISA and ELISpot are really useful for looking at a particular specific individual parameter. But multiplexing is an example of another binding assay where we can look at the detection of multiple cytokines or antibodies or antigens within a complex mixture such as serum. And multiplexing really refers to the fact that multiple things can be measured and quantified at the same time. So this is particularly good when, let's say, the sample is limited and we wish to measure multiple markers that are associated with particular disease states. So in this situation, small beads are pre-coated with our capture monoclonal antibody that's specific for a single cytokine, and these beads are actually internally labelled with a variety of different fluorescent dyes. So by mixing different beads, we can sort of analyse hundreds of different situations at the same time. This is a machine, it's called the Luminex, and it allows us to do that. So you see that we've got our beads that are specific for targeting specific cytokines, and they contain a fluorescent, different fluorescence marker. So after washing them, we then have biotinylated detection antibodies. So these are specific again for each cytokine of interest. And in this case, we have streptavidin. So we have the streptavidin PE, which binds to the biotinylated detection antibodies. So you can see that we've got this sort of situation building up, and this allows us to basically measure the fluorescence signal, but then also measure sort of the intensity. So we can simultaneously identify each bead according to its fluorescence signature, which is going to correspond to the specific cytokine, but then also measure the PE signal, which will tell us the amount of cytokine that was captured. So you can see we can start to pull various things together and measure several different cytokines at the same time. What I want to do now is move on to flow cytometry or fax analysis. So this is a picture of a fax machine. So this was a few years ago, so you can see it's quite a large machine. You've got your fluidics underneath and your computer set up there. So it's a really powerful tool for characterizing and enumerating different populations of immune cells. And the way that this works is it detects and counts cells moving through a laser beam. The cells can be identified by various markers, so you can identify T cell via your B cell, or a CD8 positive T cell versus a CD4 positive T cell. So that's useful for phenotyping cells. You can also analyze cell proliferation, look at antigen specificity using tetramers and multimers, as well as intracellular molecular analysis. So FACS stands for fluorescence activated cell sorting, but it's now often used for basically all forms of flow cytometry. So fax machines used to be quite big, but in recent years they've become more and more sort of like smaller. So you've got these small benchtop devices now, so they really have progressed. You can also measure multiple colors on different channels. Essentially what happens is you have your mixture of cells, you have your fluorescently labelled antibodies that will detect your specific cell surface markers, and these are injected into a stream of fluid so that you have cells passing through one at a time, and a laser is shone across, and you measure various different states as well as fluorescences. So in this example here, we have our cells, and we've added antibodies against different markers here. So this is an experiment to look at the basically identifying the proportion of double negative cells, double positive cells, as well as single positive CD4 or CD8 positive cells. So each individual cell is stained with a fluorescently labelled antibody that recognises either CD4 or CD8. And the laser will hit one cell at a time, and then that data will be fed back into the computer. What data you also receive back is side scatter and forward scatter. So side scatter relates to the granularity of the cell. Forward scatter relates to the size of the cell. So light scatter, it really means that it's the amount of light that's scattered, and this is affected by the size of the cell, the shape, and the optical homogeneity, and this is dependent on the angle at which the scatter is measured. So obviously, as we said before, your forward scatter is measuring the size of the cell, and the side scatter is measuring the granularity. Now, forward scatter versus side scatter is one of the first things that you will look at, and it will generally be plotted like this. So side scatter on the y-axis and forward scatter on the x-axis. And this enables you to draw gates in order to specifically identify different cell populations. So for instance, here we've run some leukocytes through the fax machine, and you can see that you start to get these distinct populations based on their size, but also their granularity. So lymphocytes are down here because they're quite small and they're not very granular, whereas your granulocytes are up here because they're larger and they're more granular. So you see you can start to draw a gate, which is literally this red line around a specific population. So when you draw a gate on the machine, you're saying, I want to analyse all the cells within that gate. So your lymphocytes will generally be found down here because they're smallest and least granular. Now, data can be displayed in different ways. So once you've got your gating data, you can then display it as a histogram. So here we've got our lymphocytes that we've gated based on their forward scatter and the side scatter. But then we want to look in this situation at how much IgD or IgM is present on the surface of the gated lymphocyte population. So what you can see here is that we've got cell number on the y-axis, and then we've got signal intensity. So we've got a peak of cells here, which corresponds to cells that are negative, so they don't have IgD on the cell surface, to a positive peak for cells that do express IgD on the cell surface. The histogram plot is useful for looking at the distribution of cells with the expression of one parameter at a time, and we can draw lines on the gate or on the plot here to actually quantify how many cells we've got in our population. So we can count the cells in the negative peak or in the positive peak by just moving the lines around on the plot. We can also express the data as these two-parameter density plots. So again, we've taken our lymphocytes here, and again, we're looking at IgD and IgM, but we have divided it into quadrants. So cells that are in this population here will be double negative, so they'll be negative for IgD or IgM. Conversely, cells up here will be double positive for IgM and IgD, and then we'll have single positive for IgD and single positive for IgM. You can also perform backgating. So normally a selected population would be gated based on the FSC, SSC. However, sometimes it's not very clear as to where the gate should be placed. And this is when it's useful to backgate, so you're confirming your gating pattern. So basically, a gate can be drawn on the fluorescence parameter and then used to set a gate back on the light scatter plot. So it's particularly useful if you're unsure of your gates or your expression levels, or whether you've identified your cells of interest correctly. That's just a useful technique. So this is looking at some FACS analysis. So here we've got quadrants. So in this experiment, we're looking at CD4 and CD8 staining of mice that have been infected with increasing numbers of S. mansoni So this is a worm infection, and we've got healthy mice here. So it's important to mention that the CD4 are stained with a different dye, so in this case PE, and CD8 with FITC. So this is red and this is green, so you can identify your different cell populations. What you're looking at is the FACS analysis of CD4 and CD8 populations in healthy mice versus infected mice. Important to have the control, which is obviously your healthy population here, but hopefully what you'll see is that with increasing numbers of the worm infection, you can see that you start to get a different difference in your cell population. So what you'll see is that your double positive, your CD4, CD8 numbers are starting to decrease, but you're getting an increase in your CD4 single positive or your CD8 single positive, and this is sort of coinciding with sort of like thymocyte differentiation and how that actually changes during the course of an infection. So you can see that it's quite a useful technique. You can also use flow cytometry to monitor cell proliferation. So in this case, we're measuring lymphocyte proliferation. You can do it in vivo or in vitro by using a stain called CFSE. And so in order to monitor immune response, it's important to follow the proliferation of lymphocytes, and you want to minimize any disruption to cell viability and function. So CFSE is really useful here because it's very good for monitoring lymphocyte division in these experimental situations. Now, CFSE is non-fluorescent until it enters viable cells and it diffuses through the cytoplasm. And then as the cell divide, the CFSE is split equally between daughter cells. So your signal from the CFSE will diminish as the cells divide and the population expands. And you can see that here. So we have increasing dose of antigen, which is causing the lymphocytes to proliferate. So you can see at a minimal dose, the cells aren't proliferating. So we have a single peak of CSFE. And then as we start to increase the antigen, you'll see that your lymphocytes start to proliferate, and we get these individual peaks as the cells start to divide and the CFSE is spread throughout the different cell populations and it moves towards the left. And then ultimately, once all the cells have proliferated and gone through, you end up with a peak shifted to the left. So you can see how you can use this stain to monitor cell proliferation. We can also use FACS to identify T-cell receptor specificity. So in this case, we can use MHC tetramers, and these are really good for the targeted detection of specific MHC peptide complexes. So the affinity of a single MHC peptide TCR interaction is too low to measure, so that's why we use a tetramer. So we have a streptavidin bead with 4 of these MHC class 1 tetramers, and they enable the signal to be amplified. And basically, this MHC tetramer technology is based on the ability of the MHC peptide complexes to recognize antigen-specific T cells at a single level. And this is really kind of a breakthrough technology that allows researchers to precisely measure the targeted T cell response to an infectious disease, cancer, or autoimmune disease. And this is kind of useful for the paper that we'll discuss tomorrow. Intracellular FACS staining can also be done for cytokines and transcription factors. So a modification of the basic FACS protocol can be used for looking at intracellular antigens. So in this case, T cells are treated with an inhibitor that blocks protein export, so it allows cytokines to accumulate. You can then permeabilize the cell, and then you can introduce your antibody, your conjugated fluorescently labeled antibody, into the cell, and then detect intracellular cytokines. And so that's particularly useful for detecting a cytokine profile at a single cell level. So in terms of FACS, as I mentioned, we can do FACS sorting. So in this case, it's very similar to FACS. You have your laser, which analyzes a particular cell in a droplet. So there's a droplet, and each droplet contains one cell. And then let's say, depending on the color, whether it's red or green, you have these deflection plates, and these are charged, and it will literally flick your cells, depending on the staining, into different collection tubes. So in this case, you can collect FITC stained cells in a separate tube to your PE stained cells, which will be in a separate tube. So that's cell sorting, where you want to actually harvest the cells, the different cell subtypes. And then another way to separate cells is magnetic beads. This is a similar process, but is much less complicated. So what you do here is you have your cells, you label them with your antibodies, and these are conjugated to magnetic beads. And then you load this onto a column that has a wire mesh, like a wire iron wool mesh in there. And then you add your antibody. You add your magnet. And what will happen is the magnet will obviously retain the cells that are bound to your magnetic bead conjugated antibodies. And anything else will be allowed to flow through. So this way, what flows through would be your negatively selected population. Then what you can do is you can remove the antibody and then allow your positively selected population to flow through. So if you remember, we talked earlier about negative and positive selection when it comes to magnetic beads, and this is what it refers to. There have also been some recent developments in flow cytometry. So one of these is the CyTOF. So this is a variation in flow cytometry in which antibodies are labelled with heavy metal ion tags rather than fluorophores. And this technology is based on detecting the heavy metal atoms coupled to antibodies, and this is then analysed. So basically it's combining your liquid fluidics, your sort of like your FACS approach, with mass spectrometry. That allows you to measure hundreds of different parameters, so it's really powerful there. But it's limited by the fact that the cells are destroyed in the process. So obviously with cell sorting, you can put your cells through the FACS machine, but you can recover them and keep them and then grow them in culture. So, some things to think about. And these are the resources for this week. So there's some particularly good reading in Janeway based on techniques, and also in Kubi as well. And then just remember that tomorrow we have a paper that we're going to go through. So this is obviously based on FACS analysis, so it'd be helpful if you go through that, and the questions are in the slide handouts for tomorrow's lecture. So just have a go and attempt some of the questions and we'll run through them tomorrow. But otherwise, thanks, and I'll see you guys tomorrow.